First of all, thank you very much for the invitation and for giving me the opportunity to present our work on oncolytic measles virus for immunotherapy of cancer, which was performed here at the National Center for Tumor Diseases in Heidelberg. So initially, the idea to use measles virus for cancer therapy emerged after clinical observations such as this one. So this boy suffered from an orbital Burkitt's lymphoma, and then he acquired measles. You can see maybe the rash here, and the lymphoma went into remission. So this was one of the first cases, one of the first hints that perhaps measles could affect cancer cells. And this has been developed further, making measles one of the roughly 10 or 12 oncolytic viruses currently under clinical investigation. So measles virus is a negative strand RNA virus. It replicates exclusively in the cytoplasm, so it does not enter the nucleus. It belongs to the family of Paramyxoviridae. It has two surface glycoproteins, hemagglutinin and fusion. Hemagglutinin mediates receptor binding to CD46, a complement regulatory protein which is often upregulated on tumor cells, to SLAM and Nectin-4. Fusion mediates a membrane fusion, as the name already indicates, and leads to the characteristic cytopathic effect of measles, which is syncytia, so fusion of neighboring cells. And for cancer therapy, we use the vaccine strain of measles virus, which has already been used to vaccinate millions of children worldwide, so it's considered very safe. So, as you've heard from Jean and Antonio, oncolytic viral therapy works on two levels. So, on a cellular level, the virus will not replicate in normal cells, but it will efficiently replicate and lyse tumor cells. But more on a systemic level, this treatment also has immunological effects. So by the viral infection, you create an immunostimulatory microenvironment through pathogen-associated and danger-associated patterns. The release of tumor-associated antigens by viral lysis kind of creates an in situ tumor vaccine, and this can lead to a systemic and lasting anti-tumor immune response. This is also the main focus of work in our lab. Currently, we would like to enhance these immunological effects. For instance, we insert a certain immunomodulatory genes into the measles virus genome, which can be engineered fairly easily. For instance, we use cytokines such as GM-CSF or interleukin-12. We also use bispecific T-cell engagers encoded in the viral genome to recruit T-cells to tumors which only have a scarce immune infiltration. We are also expressing tumor antigens from the measles virus to use it as a vaccine. And we are also aiming at combining oncolytic viruses with T-cell transfer. So measles has been developed into a versatile platform which allows for modification on several levels. So the viral envelope can be modified for targeting to tumor-specific surface molecules. For instance, you can use single-chain antibody fragments or DARPins for targeting. Also, the coding capacity of the viral genome is quite large, so we can include therapeutic transgenes in the genome up to several kb. Of course, these two strategies can also be combined, and since most people in Western countries are immune against measles, so they have neutralizing antibodies, you can modify the viral envelope to avoid antibody neutralization. This is often referred to as stealthing, or you can use different strategies such as cell carriers to avoid neutralization. So meanwhile, we have developed a variety of immunomodulatory measles viruses which act at distinct phases of an anti-tumor immune response, and I will show some of our results. For instance, we developed viruses encoding GM-CSF to enhance dendritic cell maturation and antigen presentation. We have different transgenes to support the effector immune response. for instance, interleukin-12 or bispecific T-cell engagers. And we have viruses to counteract the immunosuppressive tumor microenvironment, for instance, by encoding checkpoint modulating antibodies or co-stimulatory molecules in the virus. So our first study on immunomodulatory measles virus was with viruses encoding GM-CSF, and we studied these viruses in the MC38 immunocompetent colon carcinoma model. So what we observed here when we compared mock treatment with UV inactivated measles virus to a GFP encoding virus or the GM-CSF encoding virus, that the virus alone led to a delay in tumor progression. We only saw complete tumor responses in those mice treated with the GM-CSF encoding virus. And when we re-challenged those cured mice, they rejected tumor engraftment in contrast to control mice where we saw tumor outgrowth. So this was our first indication that measles virus can indeed act as a tumor vaccine which induces a lasting anti-tumor immune response. We analyzed this further by implanting tumors on both the left and the right flank of mice and treating only one side of the tumor, either with mock treatment, the control virus, or the GM-CSF encoding virus. And what we saw was that in the mock mice, tumors grew on both sides. In those mice treated with the control virus, tumor growth was delayed on the contralateral side, but still both tumors eventually progressed. Whereas in those mice treated with the measles encoding GM-CSF, the treated tumors went into remission, and on the contralateral side, there was hardly any outgrowth. So we wanted to analyze some possible mechanisms mediating this response. And what we observed was that treatment with measles encoding GM-CSF led to an increase in CD3 positive T cells, especially at the invasive margin of the tumor. We also extracted splenocytes from the treated mice and performed a cytotoxicity assay and interferon gamma ELISpot after co-culture with tumor cells. And we saw a significant increase in tumor-specific cytotoxic response in those mice treated with measles encoding GM-CSF, and also an increase in the anti-tumor interferon gamma response. Next, we took a different approach and developed measles virus encoding immune checkpoint antibodies. So antibodies encoding targeting either CTLA-4 or PD-L1. And we studied these in D16 melanoma. And when we compared either mock treatment with a virus encoding the antibody constant region or the anti-CTLA-4 or anti-PD-L1, we saw a significant decrease in tumor volume in those mice treated with measles anti-CTLA-4. And in case of anti-PD-L1, we saw a prolongation of survival. We also combined local intratumoral measles injection with systemic anti-CTLA-4 or anti-PD-L1 antibody in the B16 melanoma, where the antibody treatment alone only has minor effects. Here, in case of the anti-CTLA-4 blockade, we saw that combination with local measles and systemic anti-CTLA-4 is superior to treatment with measles encoding the antibody. But in case of the anti-PD-L1, both systemic administration of the antibody and measles encoding the anti-PD-L1 antibody were similarly effective. We also went further to analyze tumor-infiltrating lymphocytes, and we saw that treatment with measles encoding checkpoint antibody leads to an increase in total T cells, so this is FOXFOR CD3 positive cells, and an increase in the ratio of T effector to regulatory T cells, so indicating that the treatment led to a shift towards a favorable immune profile in these tumors. But what about tumors that lack sufficient T cell infiltration? So for this, we have generated measles virus encoding T cell engagers. So these are antibodies which consist of two single chain domains. One targets CD3 on T cells and the other a tumor-specific surface antigen. So this can really directly recruit T cells to tumor cells. The benefits are that this is independent of T cell specificity and co-stimulation or antigen presentation. By encoding the bispecific T cell engager in the viral genome, we can have localized and lasting BiTE expression. So this avoids some of the toxicities associated with systemic BiTE application, and also leads to a prolonged expression at the tumor site. So we are currently preparing a clinical trial. Of course, there are many more issues that we have to deal with aside from the preclinical lab work. Currently, we're quite far with a phase 1b/2 trial where we will combine intratumoral oncolytic measles virus in liver metastasis of pancreatic cancer in combination with systemic anti-PD-1 antibody treatment. So in the trial, there will be first a safety lead-in where we apply measles virus only intratumorally into the liver metastases at a dose of 10 to the 9 particles. And if this proves safe, we will go on to combine it with the anti-PD-1 antibody. Of course, since this is an early phase trial, the main clinical objective is safety and possible hints at anti-tumor activity. But we are accompanying this trial with a translational research program where we obtain serial biopsies prior to treatment, during the treatment, and possibly at time of disease progression, and perform our analysis of immune effectors in those biopsies. So looking at tumor infiltrating lymphocytes, at the cytokine profile, at the T cell repertoire. We also have the possibility to perform exome sequencing and identify possible neoantigens. If we have enough material, we would also like to perform co-cultures of patient tumor cells and immune cells to see if we have a tumor-specific immune response. So I guess this is a main benefit of the trial, that we possibly also gain some insights into possible mechanisms of response and no response.